Um, Hello, everyone. I'm Mila Stroganoff, your host, and on behalf of the board of the Friends of San Pedro Valley Park, I'd like to wish you a Merry Christmas, Feliz Navidad, Joyeux Noël, Sărbători Cristove, Happy Hanukkah, Happy Kwanzaa, and wishing one and all a new year filled with warmth, joy, happiness, and the worldwide recovery from the COVID-19 crisis. So, and this particular lecture is being recorded. John R. Blair agreed that we could record it, and so it will be posted on our website with our other ones, which at the end of this year, we will have 5 of our lectures, 5 of our webinars up there. And if you've missed one of those, you're more than, it's a great opportunity to go and check them out. And if you have already heard one of the lectures, it's a great time to just review something, because I always pick up something extra when I listen to a lecture a second time, or even a third or a fourth time. So that's for all of you. Oh, and the website. Yes, sorry, need to mention the website. It's friendsofsanpedrovalleypark.org. So do check us out. There's lots of things going on, things of interest that you may wish to check out besides the lectures. If you mark your calendars for Saturday, January 23rd of 2021, for 4 p.m., which is our usual webinar time, Dr. Tom Parker will be joining us, and he'll be speaking to us about manzanitas and some of the relatives, the relatives he hasn't spoken to us about, and he's very, very anxious to do so. So please mark your calendar for January the 23rd at 4. And in February, Saturday, February 27th at 4 p.m., we have Dr. Natalie Nagalingam, who is a botanist from Cal Academy. She's actually the chair, welcoming spring with a talk on a focus on flowers, the form, structures of flowers, their connection to pollinators, and much, much more. So please stay tuned. And again, Saturday, February 27th at 4, we will be sending out emails as we usually do, Adrian does, and we will remind you to sign up for these 2 events that are coming up in January and February. And now I will move on and welcome and introduce to you John R. Blair, who has been very, very kindly agreed to come and join us in mid-December when everyone is usually busy shopping and preparing for the holidays. He is here with us. And J.R. Blair received his bachelor's degree at Evergreen State College in Olympia, Washington, and obtained his master's degree at San Francisco State University in 1999, studying with the accomplished Dr. Denis Desjardins. His thesis was fungi associated with Arctostaphylos in Northern California. Since then, he has been an active member of the Mycological Society of San Francisco. He served a 2-year term as president and was the Fungus Fair chairperson for 5 years. He has taught mushroom identification workshops for mycological societies and outdoor education programs for many years, including us with bio blitzes and all sorts of things at San Pedro Valley Park. And he is currently a lecturer of biology at San Francisco State University and is director of the University Sierra Nevada Field Campus. As you might have heard too, we were chatting away and we discovered that J.R. Blair is retiring at the end of December. So he's no longer going to be the director of the field campus, and he is no longer going to be teaching at San Francisco State University. So we'll have a chance to get him back for another lecture, I'm sure, or a bio blitz in the park or something exciting. So, yeah. So, JR, take it away.
All right. Thank you very much, Mila.
You're welcome.
Yeah. Thank you all for coming today. I see some familiar names in the list there, and glad to see that you're joining us, including a couple of my students. So this is the last hurrah for them, the last chance for extra credit. But I tell you what, I'll give extra credit to all of you who are here today. So you can count on that. Let me start. Share my screen here. All right. So basically, what I'm going to be talking about today is, how do they fit into the environment? What are their life cycles and habitats and that sort of thing, right? And as we mentioned before, you're welcome to throw a question into the Q&A there. Please use that as opposed to the chat. And if it's an appropriate time, I'll interrupt my talk and answer the question. So if there's some kind of clarifying question or something like that, that's great. And I'll be around at the end of the talk as well to take questions. Without further ado, habitat, ecology, and life cycles of fungi. I want to start off by answering this question. What is a mushroom anyway? So a mushroom is a type of fruiting body, which is the reproductive structure of a fungus. Now, most people use the term mushroom to describe a specific type of fruiting body, and that has a stem and a cap, much like you see in the picture here. But there are many other types of fruiting bodies. Oh, by the way, before I go on, if you see some words in red, that's going to be on the exam, so you should make note. All right, so besides the kinds of mushrooms that have a stem and a cap, there are things like the coral fungus that we have here, or puffballs, jelly fungi, shelf fungi coming off trees. So these are all fruiting bodies. Now, some people can, expand out the term mushroom to include, all of these kinds of fruiting bodies. Other people are more strict about their definition. So I'm going to pretty much use the term fruiting bodies to describe these structures. Now, the job of a fruiting body is to disperse spores. And the definition of a spore is that it's a single cell that's capable of cell division, or germination. You could use that term, that's frequently used. Spores are microscopic. You need a microscope to see them, at least individually. About the average, for those of you who are familiar with microscopic measurements, the average mushroom spore is about 10 to 15 micrometers or microns. Now the spores divide, they undergo cell division, and they become these chains of cylindrical cells that we call hyphae. And it turns out that all multicellular fungi and a few other types of organisms are comprised of hyphae. And again, in order to see those cells, you need to have a microscope. Here's a micro photograph of some hyphae. And you can see, I want to point out that that structure, that cellular type of, the type of cells called hyphae, long cylindrical chains, creates a really high degree of surface area in contact with the substrate. And the hyphae grow into a tissue that we call mycelium. And mycelium is basically, this is the part of the fungus that is growing in the substrate, obtaining food, and essentially participating in the ecology of the environment. Mycelia from two different individuals can fuse, and that, ladies and gentlemen, is mushroom sex. We'll get into that in more detail a little bit later. And then at a certain point in the life cycle of these, this mycelium that's growing out into the substrate, the fungus then produces the fruiting bodies. Now, this point in the life cycle that can happen is dependent upon a lot of factors, many of which we are not fully aware of how it works, or maybe not even aware of the factors themselves. But for example, you can consider that a fungus that's running out of resources, so it's basically has depleted the food source, may want to produce fruiting bodies to, proliferate and to propagate and continue the species. But there are other factors like moisture, right? So fruiting bodies need to have water in order to develop. May have to do with temperature. Light seems to have an effect on all of these things. So there's a lot of factors that go into this, and it really depends upon the kind of mushroom that we're talking about. When that point comes, when the point in the life cycle, the mushroom is ready to produce fruiting bodies, it channels nutrients through the hyphae, through the mycelium, to create these dense aggregations of hyphae that we call pinheads. And oh, sorry, this is a wrong type of pinhead. Oh, there we go. That's better. So these right here are pinheads of oyster mushrooms. So this is actually a mushroom kit. What you see there is straw. The white material is the mycelium, and this kit is ready to make its mushrooms. So it's focusing all of its energy into these little pinheads, which will then grow up to be mushrooms. So now it turns out that at this stage that we see here in the front of this illustration, the vast majority of cells that are going to be in the fruiting body are already there. They're just very tightly packed. And so you can think of these things as basically swelling with water, with moisture from the substrate into the fruiting bodies, and then they become larger and mushrooms that we know and love. So the fruiting bodies, like I said, their job is to produce spores, and so they have all fruiting bodies have some sort of a fertile surface. Now that could be gills, like we see here, and all of you are familiar with gills on the undersides of white button mushrooms and portobello mushrooms and oyster mushrooms and things like that. By the way, don't confuse that with fish gills. They don't do any of this getting oxygen out of water thing. They just kind of look like them. Could be tubes instead of gills. So boletes and polypores have tubular structures within which within each tube there are spores developing. They could be teeth, like we see here on this little hedgehog here. They could be inside, for example, in puffballs or truffles, or they could just be on the exposed surface of the fruiting body, like we see with coral mushrooms, cup fungi, jelly fungi, and things like that. All right, so this is a typical mushroom life cycle. By the way, if you want to go grab a cup of coffee, this is probably a good time because it's going to get a little dense. But I'm going to walk through this with you step by step. Again, this could be on the exam, so pay attention. So let me start with the spores. So here are a couple of spores. They're color-coded red and blue. Now, and by the way, the red dot and the blue dot represent nuclei that are inside of these spore cells. Now, of course, in nature, they're not going to be red ones and blue ones, but you'll see why we put those there, or why the illustrator put those there. All right, so the spores are dispersed from the fruiting body. They go out into the environment. Perhaps they're caught up in a wind, a little bit of wind or a little breeze here and there. There are some spores that are even delivered by animals. They get caught up on their body parts and are moved from one place to another. In any case, they're spread out. Now, if they land in the right place and the conditions are right, enough moisture and light and the temperature is correct and all that sort of thing, then they'll germinate. They'll undergo mitosis to create the hyphae. All right, so here's those chains of cells. The blue one does it and the red one does it. Now, if, again, depending on the conditions, if everything works out well, two different hyphae will come together and fuse together. And this is what's called cytoplasmic fusion. Now, again, like I say, everything needs to be right. So here's where the color coding comes into play. They need to be mating types that are compatible. So for example, with animals and with plants, there are just two mating types, as male and female, right? Turns out with some fungi, there are as many as 100 different mating types. But if the mating types are the same, they won't fuse, right? So just like, again, taking the example of animals and plants, two sperm cells cannot fuse together and two egg cells cannot fuse together. But if they're a different mating type and compatible, they will fuse together to form a different kind of hyphae or different kind of mycelium. Each of the cells, the hyphal cells, now have both a red nucleus and a blue nucleus. Now, those of you who remember your biology, with plants and animals, humans, for example, when a sperm cell fuses with an egg cell, almost immediately, right after that fusion of the two cells, there is nuclear fusion. So the nucleus from the male fuses with the nucleus from the female. You remember the terms haploid and diploid? Remember those? So haploid means that there's just one set of chromosomes in the nucleus. Diploid means there's two sets. One came from mom, one came from dad. And that creates the diploid cells. Now with fungi, or at least most fungi, this doesn't happen right away. And so if you look at all of these hyphal cells in the lower part of this illustration, you'll notice that there is a red one and a blue one. That means there's two haploid nuclei that do not fuse, at least not for now. Later on it'll happen. I'll get to that part. Okay, so, and by the way, that's the abbreviation for this would be N plus N. Whereas if it's a diploid, it would be 2N. If it's haploid, it's 1N. Right. Okay, so the hyphae is growing, the mycelium is growing around, and it's, doing its thing. It's eating material in the substrate. It's releasing enzymes into the substrate, which is really important ecologically. I'll get back to that a little bit later. And then the conditions are right for it to produce the fruiting body. And again, it channels nutrients to certain places to create the pinheads, and then fills with water, and then we've got the mushroom. We got the fruiting body. Now, on the fertile surface of that fruiting body, the gills, for example, in this illustration, there are certain terminal cells. These are cells at the ends of these chains of hyphae called basidia. And in the basidia, in the primordial basidia, early in the stage, there again are two nuclei, one from each of the parents. At this point is where nuclear fusion happens. And this is where it goes from being haploid to diploid. Now, it turns out that the diploid stage of these kinds of fungi is very short. It doesn't last very long before meiosis happens, which is a conversion from the diploid stage to the haploid stage. Typically with meiosis, what the typically the result of meiosis is four haploid cells, four haploid nuclei as a result of meiosis, which then, in the case of fungi, these haploid nuclei, you can see there's four right there, migrate out to the tips of these little finger-like structures to become spores. Now, there's exceptions to all of these things that I was talking about. So, for example, the store-bought mushroom, your white button mushroom, which is actually the same species as portobello, and it's also the same species of mushroom that we call crimini. Those are all just variations. The standard name, the Latin name, is Agaricus bisporus. So there are only 2 spores on each of these basidia, as opposed to 4, which we see in most fungi. Chanterelles, many of you are familiar with those, will actually produce 8 spores on these basidia. In any case, out of the spores form at the ends of these little finger-like structures called sterigmata, and they are released and the whole cycle starts over again. Okay. All right, if you're still out getting coffee, come on back. I'm done with the dense part. All right, so let's talk about the importance of fungi in the environment. So there are 3 basic categories within which fungi fall. They can be decomposers. They can form mutualistic relationships with other organisms, or they can be pathogenic. So they can, pathogenic, so they can be diseases, right? All right, so let's take each of those one at a time. Let's start with fungi as decomposers. So about half of, I should change this actually a little bit, about half of the fungi that we see as mushrooms are saprotrophic. It turns out a lot larger percentage of fungi in general are saprotrophic. Saprotrophic means literally dead eaters, right? Dead eaters. Sometimes you'll see, especially in older literature, you'll see the term saprophytic, but that really doesn't make sense because sapro means dead and phytic means plant. And it isn't always plants that fungi are decomposing. It is most of the time, you know, wood and leaves and things like that, but it can be other things as well. So saprotrophic is a much better term to describe this. So here's some statistics. It turns out that about 50% of the microbial biomass, so this is, we're talking about microscopic organisms, about 50% of the microbial biomass in compost and in grasslands is fungal. And it turns out the other half is bacterial in this case. And in forests, it's even higher. So somewhere between, estimates of between 60 to 90%, depending upon the forest, of the microbial biomass in the soil is fungal in nature. And saprotrophic fungi include things like litter decomposers, like we see this troop of mushrooms here on the in the upper photograph. And actually, you know, I think I may have taken this photograph at San Pedro Valley Park. And then here's oyster mushrooms growing on a log. The term lignicolous, by the way, means wood-loving fungi. So it turns out that oyster mushrooms are wood lovers. All right, let's talk about fungi as mutualists. So this mutualism is where there's 2 organisms that are both benefiting from the relationship. The type of relationship that fungi participate in that is mutualistic is called mycorrhizal. Let's break that word down real quickly. So myco means fungi, rhizae means roots. And it turns out that over 90% of all plant species have this relationship. Think about that for a minute. Over 90% of all plant species have are mutualist, have a fungal partner that's mutualistic. And it turns out that in this relationship, that the plants provide photosynthetic products for the fungus, sugars basically, and fungi are involved with the conversion of nutrients such as potassium, phosphorus, and nitrogen into usable forms for the plant. So the fungi are really important for the plants to get their nutrients. And in some cases, there's also an enhanced water uptake as a result of this fungal partner in the relationship. By the way, there are two basic types of mycorrhizal. This one is called arbuscular mycorrhizae, and this is an arbuscule. Most mycorrhizal fungi are arbuscular mycorrhizal. And then the other type is this one called ectomycorrhizae, and a few of them are that way. It turns out that most of the fungi that produce, the mycorrhizal fungi that produce fruiting bodies are ectomycorrhizal, and there's only a few that are arbuscular mycorrhizal that produce fruiting bodies. All right, so a lot of your familiar edible and poisonous fungi are ectomycorrhizal. So let me run through these pretty pictures with you real quick. This is a bolete from the Rocky Mountains. Really a beautiful specimen, and it was really delicious too. This is a white chanterelle. This is a deadly mushroom here that's from the East Coast. This is Amanita bisporigera, and then this is another edible species here called matsutake. So really highly sought after mushroom in Japan. All right, another category of fungi of a mutualistic fungal photosynthetic partner relationship are lichens. And it turns out that lichens are either a relationship between a fungus and either an alga or a cyanobacterium. The photobiont, which would be the alga or the cyanobacterium, provides, again, photosynthetic nutrients for the fungus. And the mycobiont, the fungal partner, provides a distinctive home for the photobiont. So if you think about algae, for example, and cyanobacteria, for that matter, are typically found in aquatic environments. So they're in water. But by forming a relationship with the fungus, they could live on trees or on rocks, fence posts, old cars, lots of different things, right? Now, and by the way, just to kind of show you here, this is a fungus that has little fruiting bodies. So these are little ascomycete fruiting bodies. This is a micro photograph of a lichen. The red or the purple colors are fungal cells, and the green colors are little pockets of algae. And this is stained so you can see those two different types of organisms. So this is a mutualism, or is it? Now there's a lot of argument about this, and there are some people who will argue that it's not really a mutualism, it's more of a parasitism, and that the fungus actually has the photobiont captive and has locked it up and is basically taking food from the alga or the cyanobacterium. One of the points of evidence of this is that the species of algae and cyanobacteria that form these kinds of relationships do just fine without the fungus. We find them growing, usually in aquatic environments, but we find them growing elsewhere without the fungus. But the fungi require the cyanobacteria or the algae that are parts of the relationship and can't live without them. All right, let's talk about fungi as parasites. Well, of course, we are all familiar with that as human beings. Athlete's foot and yeast infections, those are both parasitic fungal infections that we experience. But let me go beyond that and talk a little bit about fungi in other as diseases for other organisms. So a lot of fungi form diseases on plants. So Dutch elm disease, and this is an elm right here that is succumbing to that pathogen, and chestnut blight, both are fungal diseases that have really changed the way that the eastern woodlands look. If you were to go back 300 or 400 years before these diseases were introduced to North America, you would see very different kinds of forests that include the elms and chestnuts. Here in California, many of our Monterey pines are suffering from pine pitch canker, which is a fungal disease. If you drive down the coast here in San Mateo County, you see just acres and acres and acres of dead Monterey pines that have succumbed to that disease. That also is an introduced disease. And then there's one species of mushroom, both of these, all three of these diseases, so the Dutch elm disease, chestnut blight, and pine pitch canker don't produce fruiting bodies per se. They reproduce asexually. But there is a mushroom called a honey mushroom that can cause root rot in trees. Just a little anecdote, I was working a fungus fair one year, and two women approached me, one mother-daughter pair, and they had a little paper bag, and they put the paper bag in my face, and I looked in there and I see honey mushrooms, and they said, "So we have these things growing in our yard. What are they?" And I told them they're honey mushrooms, and I said, "Are they near a tree?" And they said, "Yeah." The mother said, "Yeah, my daughter here planted this avocado tree when she was a little girl, and these mushrooms were growing at the base of it." And I felt like, a doctor who was basically telling a family that they had a loved one that was going to, well, you get the idea. It was a sad moment, I have to say. Anyway, honey mushrooms are actually an edible species. A lot of people eat them, but they are parasitic on trees. There are some fungi that also parasitize other fungi. So for example, many of you are familiar with this mushroom here. It's called the lobster mushroom. It's actually a really good edible, has a really nice subtle flavor. Well, it turns out that it is actually a parasite that's orange in color that's growing on another mushroom. And the other mushroom that it grows on is basically a flavorless, sometimes called insipid species, but it's not toxic. But when this fungus grows on there, it actually gives it some color and some flavor. There's another kind of fungus that really is interesting. It's called Puccinia, that grows on the tips of Brassicaceae plants, Arabis. And when it grows on those tips, it forms this yellow mass of spores, and then it triggers the plant to actually extend the tip of that plant extends, it grows a little bit longer. And so look at this thing here. So here's the Brassicaceae plant. Here's the extension. There's the yellow spore mass. And look, there's a little insect, it's a little wasp that has landed on that thinking it's a flower. And it gets these spores all over its legs and so on. Then it flies off. And hopefully, for the fungus anyway, it lands on another Brassicaceae plant of the same species. And again, here we go again, forming these little flower mimics, if you will. Right. My understanding too is that it actually will also cause a little bit of leakage in the phloem, which is where the sugars are being transported in the plant. So it makes it even more attractive to the insects because it's a little bit sweet. All right, and then there's some fungi that are parasites on animals, or in this case, the cases I'm going to show you, are they really predators as opposed to parasites? So the genus Cordyceps, actually they've changed that now, it's called Ophiocordyceps, has members that kill insects, and then they grow out of the mycelium grows inside the insects, and then they grow fruiting bodies outside of the insect. So here, for example, this is a beetle grub, and these things that are growing outside of this beetle grub are the fruiting bodies of the fungus that has parasitized this beetle. And then there are some fungi that actually trap nematodes. So if you're familiar with nematodes, these are little worms, sometimes microscopic, many of them are. Some are not, but many of them are microscopic. And they're actually, it's a very diverse group of animals, sometimes called roundworms. And if you look at this, this is a scanning electron micrograph of a nematode right there that has ended up going into, see this little hoop right here? There's one there, there's one here, and one in the foreground. Those are hyphae, hyphal little loops made up of 3 hyphal cells that form a circle. And if the inside of that loop is touched, the fungus sends water into those 3 cells, and they swell up and trap the nematode. And then they eventually break down the nematode, and that's what they eat. In fact, many of you are familiar with oyster mushrooms. They're one of the ones that eat nematodes as a predator. Parasitic fungi actually can be beneficial. So they are frequently used for biocontrol. So here's a couple of photographs from a website I found. I think this is an acacia, I'm not 100% sure, down in Australia. That was a problem. They found this fungus that was specific to that plant that will help control the invasiveness of that pest. And then here is a beetle that is also being covered with a fungus that is used, the fungus is used as a biocontrol for that invasive species. Some plants have turned the tables though. So there are some plants that are chlorophyllous. In other words, they don't have chlorophyll. And it turns out that all orchids also have this habit of being mycoheterotrophic epiparasites. Now that's going to be on the oral exam, so pay attention. Just kidding, won't be. Yeah. It'll be on the other exam, but not the oral exam. Anyway, mycoheterotrophic parasites are, epiparasites are plants or fungi that basically parasitize a fungus, right? I'll get to what that is in just a minute. I just want to talk about these pretty pictures again. This is called ghost pipe. It's not found in California, but it's pretty common throughout the rest of North America, in northern parts of the Midwest and in the East Coast and so on. And this is a achlorophyllous plant. This is the fungus called Dracula. Some of you are familiar with that. And I would just want to throw this in here because the labellum of the orchid looks like a mushroom. Whoa. Now it's not real clear why. There's some evidence that there actually are some of the seed dispersal of this plant is executed by insects that think that this is a mushroom, and that there's some compounds that are released from this orchid that mimic the mushroom odor. Yeah. Microheterotrophs, which are fairly common, there are a number of species of common microheterotrophs in California, mostly up in the mountains. So for example, here's a rain orchid. Now you'll see that this one is green. So it's a mature orchid that is actually photosynthesizing and making its own food. But when it was young, it needed to parasitize a fungus in order to get to this point. There are a few orchids, as you probably know, that are achlorophyllous. So ghost orchid, phantom orchid, coral roots, and so on. And then here in the middle, that's snow plant. And then this is pine drops. So snow plant, pine drops, ghost orchid, those are in the Ericaceae family in the subfamily called Monotropoideae, which are the achlorophyllous ericaceous plants. All right, and then there's some categories that don't really fit into these other nice clean categories. You know, the parasites and the mycorrhizal fungi, the mutualists, and the saprotrophs. So, for example, it turns out that many plants, most plants, I think we're finding, have fungi called endophytes that are actually living inside of the leaves of these plants. So here's a micrograph where they've actually stained the fungi, the hyphal cells, blue. So all those blue thread-like things, those are fungi. And then if you look closely, you can see the cortical cells of the leaf, the mesodermal cells actually, mesophilic cells, which are the leaf cells. You can see as sort of a pale green, you can see the outlines of them. Now, this relationship is not clearly understood. There's some evidence that some of these endophytes actually produce alkaloids that reduce herbivory, so they don't taste good to the grazers. This is an ad from a company that produces a turf grass that it has endophytes over here on the right and no endophytes over here on the left. So there's some benefit perhaps for growing that, but it doesn't seem to hold true for all endophytes. One predominant theory is that they're just hanging out, waiting for the leaf to die, and once that happens, then the leaves have then this fungus has a leg up over any other fungus that comes in to try to decompose that leaf. So anyway, it's kind of a mystery. All right, and then of course there's the other way around. You know, fungi as food for animals. So for example, here's a morel, a really tasty edible species. There are a lot of mammals that have developed a taste for mushrooms. So deer and a lot of rodents will eat fruiting bodies, will eat mushrooms that are growing on the surface above ground. So for example, here's a pseulis that's had a big bite taken out of it, perhaps a rabbit, perhaps a deer, perhaps a rodent, not sure. And then mushrooms that grow underground, so truffles, basically, or false truffles, there are a lot of those. Those have evolved strong odors. And those strong odors attract rodents or pigs or some other mammals to dig them up and eat them when they're mature, and the spores then pass through their gut and are dispersed that way. This is a squirrel at the San Francisco Botanical Garden that was using one of the posts, designated plant posts for its truffle gourmet event. There are also some arthropods that exclusively eat mushrooms. A lot of insects like fungus gnats, fungus beetles. Here's this is called a pleasing fungus beetle here in the center. These are some springtails over here on the right, and this is a fungus gnat on the left, and also some other arthropods that are not insects like mites and arachnids and things like that. So, and then there are some insects that have also evolved an agricultural relationship with fungi. So leafcutter ants, for example, they don't eat the leaves. They basically collect little pieces of leaves, to feed their fungus the fungi that they're growing in their nests. So they eat the mycelium that grows on the leaves that they collect and bring down into their nests. This down here, this is an ambrosia beetle, which is a group of beetles that are severe tree pests. They're bark beetles. They weaken trees. They create real problems with trees throughout certainly in the Western North America, including the Sierra Nevada. But they also eat mycelium. And what they do is they actually have little pockets at the base of their head which they stuff with mycelium if they are moving from one tree to another. And then they basically plant that mycelium in their galleries underneath the bark, and then they eat the mycelium as it grows. Pretty cool. And then, of course, let's not forget perhaps the biggest fungus eater of all, Mark Lockerbie. Well, people in general. So here's Mark actually frying up some chanterelles after a foray at Mendocino woodlands. And here's someone with some Leccinum here, a nice porcini right there, and some a big cauliflower mushroom right there. All right, well, thank you very much. I really appreciate you being here today. And I want to thank the friends of the San Pedro Valley Park and for inviting me to speak. And I'm happy to take questions in the Q&A. We can't hear you. Oh, sorry, somebody else. That's from before.
So we'll give folks a few minutes to try and pose some questions. And Jared, thank you. That was it was fascinating, actually.
Thank you. I think it's fascinating.
Let's see.
Oh, you know, so gardening with mycorrhizae. So I'm glad you brought that up, Michelle. If you go to a nursery or a hardware store, you can find little packets of mycorrhizae. It's labeled as such, mycorrhizal mushrooms or mycorrhizal fungi. And I have to say, I'm a bit skeptical about the efficacy of that approach. It turns out that there's mycorrhizal fungi in all but sterile soil. So unless you've, you know, perhaps if you're only growing your plants in soil that you purchase from the hardware store, potting soil basically, that you may, you know, in that case, if it's sterile, you may not get very good growth because the plants are not accessing mycorrhizae. But if there's native soil in your garden, there's going to be mycorrhizae there, and your plants are going to find those mycorrhizae. So you really, I'm skeptical about spending money on these little packets of dried material that contain bits of fungi that may or may not be viable. So, so it's there.
And mushrooms per se, they don't require very much, the spores, they don't require very much moisture at all in order to start doing their thing.
Yeah, so so a lot of times, you know, if the plant is living, there's going to be mycelium in the substrate that's doing fine. It's really, so this kind of actually goes as a segue to Sharon's question about will you be seeing mushrooms at the park? You know, if there's not rain, there's not going to be very much in the way of mushrooms. Normally, the Mycological Society has their mushroom fair this weekend, and actually last weekend. Sorry, it would have been a bust. It would have been terrible. because there's just not been very much rain. We would have had to send people up to the northwestern part of the state and into Oregon to find mushrooms. So around here, it's just not going to be very good because of the drought conditions that we've had so far. Now the mycelium's still there. It's still doing its thing, and it can do quite well, as Mila suggested. It can do quite well even in dry times of the year. Now there is a limit, because at some point the mycelium is going to be ineffective if it can't grow, and it actually does need, all living things need water, it's to some extent. But the mycelium can do quite well with low amounts of water, and then once the rains come, that's when we see the fruiting bodies forming.
But the spores need water in order to actually
Germinate, yeah.
Germinate,
so again,
right?
all living cells are going to need water to some extent, but it doesn't, it could be, there's spores out there that are doing their thing right now, germinating because they're microscopic and they are not going to require much in the way of water. So, there may be, there's definitely growth happening.
Well, someone must have heard that we were going to have a mushroom talk because there was a real storm and rain.
Yeah.
Rain coming down in buckets yesterday. So
yeah, that was really
in preparation to see all the mushrooms that will pop up soon.
Yeah.
Yes.
So this time next year, hopefully we'll get more rain, and please come to our fungus fair. It's really great. I'm actually wearing a shirt from a fungus fair a couple of years ago. There's California with the mushrooms growing around it. And one of my favorite t-shirts from a fungus fair. But in a good year, it's really pretty spectacular what we see out there.
Well, we've had mushrooms in our garden, but in our courtyard area, but of course, not as many.
You've been watering.
Huh?
You've been watering, right?
Well, we've been watering, but we always seem to, yes. But we have quite an array of various fungi that pops up here and there, and sometimes it's like it seems to move, like brings up things as though it's coming up, like I don't know what it is, but it just literally, it's like a little volcano.
Yeah, send me a picture.
Yes, well, they didn't pop up this year, but they did, in the previous years, and I thought, oh, I was waiting for it to take a picture because it literally lifts stuff and it's like a little wall, and I'm going, good gracious.
Yeah.
All right.
So there actually are some mushrooms that can actually go through pavement, so asphalt or concrete.
That's it. That's it.
Simply from the persistent, but it's small but persistent pressure. It can pop up through that sort of thing. I have a couple more questions here. So Anonymous asked about some small brown mushrooms growing on eucalyptus bark. And that could very well be from heavy fog if there hasn't been any rain. That is actually a source of moisture for a number of mushrooms. We have fog drip mushrooms, if you will, that grow in the summer in coastal California. So that can be a source of moisture. And of course, with eucalyptus trees, they hold the mushroom moisture in their bark because of the sort of fibrous nature of the bark, right? Sandy asked about how fast do mushrooms grow? It really depends on the mushroom. So, something like, I kept this on purpose. So like, for example, see these mushrooms right here, these little litter decomposers can basically show up overnight. Sort of the most famous kind of mushroom for growing really fast are the stinkhorns. They can basically go from the egg, they form an egg. They can grow from an egg to being a foot long in some cases in a few hours. But on the other hand, something like this right, whoop, sorry, that right there, that's a polypore, a shelf fungus. And some of those actually will take, can grow for decades, basically, and get bigger and bigger every year. So they're perennial in nature. And then let's see. And then, so how soon will mushrooms show up? Again, it's interesting because it's not an exact science, and there's not a good way to answer that without saying, well, it depends. It depends upon a lot of other factors. And I've noticed that just anecdotally, and this is not something that's necessarily scientifically proven, but I've noticed that there's some mushrooms that seem to give up. So if typically they're a November, December, something we find in the late fall, and there's no rain until December or January, that we don't see them until the following year. So that could happen too. Hey Bing, how are you? Yeah, that's a really good point. In fact, if you look at, so being asked, why do some polypores look to be sweating? So you can see this one in particular. This is called the red-belted pine conch, and it's kind of famous for that, for producing these droplets, moisture droplets on the fertile surface, which is the white part underneath. And it's actually, I used to think that it was just sort of condensation, and that it's just really structurally good at grabbing moisture out of the air. But it turns out that it's actually coming from the substrate. Hang on just a minute. I want to turn the light on. It's getting dark. There. Now we can see me again. So yeah, so that's called guttation, I think, is the actual term for that. And we see a lot of polypores doing that, particularly that species.
And do you consider lichens to be parasitic?
No, they're not. Yeah, they're commensal, which is I didn't talk about that, but so there's basically three kinds of symbioses. There's mutualistic, where both benefit. There's parasitic, where one benefits and the other one is at a disadvantage. And then commensalism is where one benefits and the other one is not affected. And so lichens, I would consider to be commensal. They can grow fine on trees, or but they can also grow fine on fence posts or rocks and things like that. But they're not stealing from the plant. Some people think that that lichens are actually overtaking the foliage of like a tree, right? So if you look at oak trees deciduous oak trees during the winter time, it looks like that they're just covered with lichen, and how can they grow new leaves that way? Well, it turns out they do fine, unless they're already diseased from something else. And in which case, the lichen actually just takes advantage of the fewer leaves. But a healthy tree will not be affected by lichen.
Well, that's good.
So Anonymous said some Amanita species are deadly to us humans. Are they eaten by other animals? And if so, what adaptations do they have that humans don't, allowing them not to be killed by the toxins? Okay, so yeah, it's simply a matter of physiology. That there definitely are certainly insects that can eat Amanitas. If you find a death cap, which is the most common deadly Amanita in California, and you find one that's getting a little old, you'll find springtails and fungus gnats in the mushroom. The death cap's a little unusual because it's introduced from Europe. So I don't think there are any mammals that eat Amanita, but there are mammals that will eat other members of the genus Amanita that are deadly to people. And I think it's just a metabolic system that allows them to metabolize the amatoxins that are in there. I'm not a physiologist, so I don't know any more details than that. So.
But in human beings, the liver is usually destroyed.
Well, it turns out that that the amatoxins, what they do is they disrupt RNA synthase. It's an enzyme, RNA synthase, and so if you RNA synthase is really important for making proteins. And so if proteins are not made, then cellular function ceases to, it just stops working. So any cell that is in contact with amatoxins that are intact and not metabolized, those cells stop working basically. And the reason that our liver is destroyed by these toxins is our liver cells are constantly reproducing. And if they can't, in the liver, right? Just the liver. And so if the liver cells can't reproduce, then it atrophies and we die from liver failure. But it's actually doing the same thing to like the lining of our gut and our kidneys and all kinds of other organs, but it's just the liver that fails first.
Ah.
Anonymous asks, what is a toadstool? And that's kind of like asking what is a mushroom, right? It depends upon who you talk to. So there are some people that will talk about anything with gills is a toadstool. Other people say anything that's poisonous is a toadstool. So it doesn't have an actual definition.
And are there bioluminescent mushrooms?
Oh, yes, most certainly. I don't have any pictures of them right with me, but so it turns out that with bioluminescent fungi, which are most diverse in the tropics. So if you go to the jungle, go to the tropics, and take a night hike, and the best thing is to have good night vision. So you want to have you want to be in the dark for a while so your pupils expand, so dilate. So if you have well dilated pupils and you take a night hike in the tropics at night without using a flashlight, dangerous, but you'll see them. You'll see them everywhere. Some places it's like you can almost see where the pathways are because they're lined with the bioluminescent fungi. Now, sometimes it's the mycelium that's bioluminescent. Sometimes it's the whole mushroom. Sometimes it's a part of the mushroom. There's a lot of variation here. Now, here in California, we have a couple of sort of famous bioluminescent fungi. One's called the jack-o'-lantern mushroom. And the jack-o'-lantern, actually, the whole fruiting body will glow in the dark, but it's not very strong. So you have to have like complete night vision in order to see it. And that takes about a half an hour for your eyes to fully dilate. So here's my suggestion. If you find a jack-o'-lantern mushroom, collect a fruiting body and put it on your bedstand and set your alarm for the middle of the night. And you're guaranteed to have fully dilated pupils and you'll be able to see it glow. It's pretty cool. And then there's the armillaria, which is a honey mushroom. The mycelium glows, and some of you know that as what they call firefox, I think. Anyway, there you can see little glowing bits in the dirt, in the duff, in like redwood forests and so on. If you, again, if you have good night vision.
So this is bacteria that's causing, nothing to do with bacteria.
Oh, it's the mushroom actually. So yeah, but it's the same pathway or similar pathway anyway that's seen in fireflies or bioluminescent phytoplankton or undersea fish and things like that. So it's basically it's a luciferase is the enzyme that's breaking down a compound called luciferin, and it emits photons. And it's actually the organisms themselves that are doing that. Now, some people say, well, why does it do that? Why is it doing that? Well, that's not clear. There's a lot of speculation. So there's some minimal evidence, although it's not really well established, that some insects are attracted to bioluminescent fungi. So they think there may be a spore dispersal mechanism there. But then there's other fungi that that doesn't seem to apply to, that are bioluminescent. So it's one of those things. It's kind of like, I mean, if you look at, if you look at this picture here, for example, why is that pink? Why is that slimy? Why does this have little cracks? Why is this, why does this have water coming out of it? And you can almost answer it the same way for anything until we know better. And that is, well, it's just an accident of how these things have evolved and how they grow and how they metabolize and so on. And there doesn't seem to be a lot of value, of evolutionary benefit for a lot of these characteristics. Same thing with being very flavorful, or being bitter, or being toxic. So with a lot of things, you can say, for example, a plant being toxic or tasting bad is to prevent herbivory. But with a fruiting body, when it's gotten to this point, all of these things, it's already distributed millions of spores in some cases. It's certainly hundreds of thousands of spores have already been spread. So it doesn't really matter if anything eats it now. So that doesn't work. And there's some evidence of certain characteristics facilitating spore dispersal, like truffles, right? Smelling strong,
Mm-hmm.
or stinkhorns attract flies, and they disperse their spores that way. But most of the time, there's really no good evolutionary explanation for these characteristics. Yeah.
All right.
So Sharon asked about consistency of conditions. By the way, I am not in a hurry to leave, so I know that you set a 10-minute time limit, but if people have further questions, I'm okay with sticking around for longer. But anyway, Sharon asked about given consistent conditions and enough moisture, do mushrooms tend to grow in the same area year after year? Yes and no. Again, depending on the species. For example, when we talk about questions like this, where do we get most of our information? People going out looking for edible mushrooms, right? So some mushrooms, like chanterelles, for example, let me go here and show you a chanterelle. So here's a chanterelle. It turns out that those are really, really consistent. And so people have their chanterelle patches, and they will do almost anything to keep people out of those. In some cases, it's sort of like, if you ask, okay, where'd you get those chanterelles? Oh, I got them in California. So, and that's as best as you're going to get, right? Because they have their patch. But porcini, for example, are not that faithful to growing in the same place. So now they're mycorrhizal, so they're going to have to grow with a particular type of tree or shrub. So you're not going to find them growing like in grassland or something like that, which doesn't have the same mycorrhizal relationship. But they're not as faithful to a single place as chanterelles are. So yeah, it sometimes yes, sometimes no.
So the fact that people actually go out and seriously go about collecting and gathering mushrooms in a particular area year in and year out does not diminish the likelihood of the mushrooms coming up
Right.
every year?
So yeah, that's kind of another aspect of the question is, does it affect the organism, the mushroom, the fungus, if you go out and collect it all the time? And it turns out no. They've done a study with chanterelles, and this is really helpful because chanterelles, grow in the same place year after year. So it's easy to kind of set up an experiment like this, where they actually grew mushrooms in plots for 20 years, I think, where they had 3 different treatments. One type of plot, they left them alone. Another category of plots, they picked them. They just pulled them out of the ground. And then a third category of plots, instead of just pulling out of the ground, they cut them off, so they left a little bit of the base in the substrate. No significant difference. So every year, all 3 of those plots produced mushrooms in basically the same amount. In fact, it turned out, it's not significant, but it turns out that on average, the plots where the mushrooms were picked, either one of them grew mushrooms, more mushrooms than the ones where they weren't picked. Now, on the other hand, they did a study with this species right here. This is the Matsutake, and Matsutake, the best ones, the ones that get the most money, are still underground. They haven't popped out yet, and so people actually use rakes to try to find them. And what that does is that dehydrates the substrate, and so the next year that substrate will not be as good because it's been dried out. And also the underground mushrooms haven't dispersed their spores yet. So it would basically take them growing up and looking like this before they disperse their spores. So you can see there's 2 categories there. One, the chanterelles do just fine. Matsutake, not so much, and it's because of the way people pick them.
Mm.
Okay, so being asked about fires. Well, I have to say that I'm not up to speed on this because there's a lot of new information that's happened over the last 10 years because of the fires. But yeah, there's, it kind of depends upon the type of the nature of the fire. So do you remember the rim fire, the one that happened just outside of and into Yosemite a little bit? That fire was particularly hot. And so it turns out that the damage that was done to the soil was, 3 or 4 inches deep, which is about as maximum as you can get. Even superheated soil doesn't about 3 or 4 inches down, it doesn't affect. But it was deeper than you see with most fires. And so as a result, mycelium was basically wiped out in a lot of places in the rim fire. And they know that because they had plots. So researchers had plots where they'd been looking at the mycorrhizae before the fire came through. Then they were able to compare. Going back even further, you remember the Inverness Ridge fire? Oh, that's not what they called it. The one at Point Reyes. That was back in the 80s.
Yeah.
There was actually the same thing where Dr. Bruns, B-R-U-N-S, at Berkeley, had plots in the Bishop Pine forests at Point Reyes, and when that fire came through, he was able to do some comparisons. And so if the fire's not super hot and it moves quickly, then it's not going to affect the mycelium at all, virtually. But if it's really hot and burns in one place for a long time and gets deep, then that's bad for that. So, by the way, some people think that it's not really Dr. Bruns, it's Dr. Burns.
How deep does one find mycelium? I mean, is it
It's usually mostly pretty close to the surface.
quite close?
But as you go deeper, it diminishes. Yeah.
Oh, it does. It doesn't increase. It actually, ah.
Yeah, most of it's in the first 3 or 4, maybe even 2 or 3 inches. Most of the mycelium. So yeah, that's where the food is, right? So that's where the most of the organic materials that the mycelium are ingesting is found, is in the top layers.
Because when I've been planting various plants and digging holes for it, all of a sudden you see all of this white mycelium, and it's like, it's just literally filled with it. You
Yeah.
know, and you're going, wow, this place is busy, isn't it? With
Yeah. So there might be something going on there where there was like maybe a log or something like that that broke down there many years ago, and there's still a lot of nutrients there. But the vast majority of mycelium is going to be close to the surface.
Mm.
And a lot of times you don't see that because it's dispersed, right?
Right.
Yeah, the photograph that I took of the mycelium, that one, so that was under a log, and that's pretty typical, where you turn a log over, or if you have a piece of lumber or something that's been sitting over the winter, and you lift it up, and you'll see that.
And we have had more mushrooms in the Boletus family popping up near the oak tree. I mean, they are happy. They
Yeah.
keep popping up here and there. Yes, yes.
All temperate Boletaceae are mycorrhizal. So there are some that are saprotrophic in the tropics. Actually, there are some members of that family that can go either way. They can be saprotrophic until they find the right host, and then they become mycorrhizal. But the ones in your yard are going to be mycorrhizal. So you have an oak tree, or more than one?
More than one.
Yeah. Do you have pine trees also?
No. Well, further down, yeah, not exactly very close, but the main thing we have are oaks.
Yeah.
Yeah.
So when I was doing my my thesis dissertation, actually it's not a dissertation. I don't have a PhD. When I was doing my thesis, which was mushrooms associated with manzanita, I did my level best to try to find pure stands of manzanita. So I knew, at least for the mycorrhizal species, so I knew that they were associated with the Arctostaphylos manzanita and not with something else. Somebody told me that if there's a pine tree that's 100 feet away, it could have roots where I'm picking.
Really?
Yeah.
Wow.
So yeah, and those roots could have the mycorrhizal species growing on them.
Ah.
Yeah.
Well, it could be some pine connection then too, but they're certainly growing very close to the oak trees. So
yeah, chances are that's where they're coming from, because oaks are another group of another plant that has the ectomycorrhizae relationships. Most conifers and oaks, all oaks, a lot of the Ericaceae.
all right. Okay. So
Right.
have you answered everyone's questions?
Looks like I have.
Whoa, super.
Anyone else want to repeat or ask me any other questions? Last chance.
Yes, any clarification before we let JR go? Until next year.
Until next year. Yeah. All right. Maybe I'll talk about mycorrhizae on our next talk.
Right. Well.
And I look forward to doing the BioBlitz again.
Well, that would be lovely.
Yeah.
It would be very interesting to do once again. So thank you. That was most impressive. Most impressive. I mean, I've learned about spores and this and that and the sporophytes and all of that, but it was really fascinating to hear it with the mushrooms because we didn't cover fungi. So yeah,
Yeah, you're very welcome.
fascinating. Thank you. And
Okay, bye
thoroughly enjoyed
everybody.
it. Okay, ta-ta.
Ta-ta.